Buenos días. Yo tengo el deber de presentar al profesor emérito Giulio de Artis, que nos va a presentar en estos momentos. Él es profesor emérito y nos va a presentar ahora dónde estamos con las mucopolisacaridosis. Come on.
Buenos días. I would like to thank the Asociación MPS Lisosomales España, who invited me to this ceremony to celebrate the 20th anniversary of the association. Jordi Cruz, the director, on behalf of the association, gave me a task that was very useful for me to review memories of my past in the field of metabolic diseases and of lysosomal diseases in particular. Actually, at the beginning, he asked me to talk about my experience in 20 years through lysosomal storage diseases, and then we discussed, and the title, as you see, was changed in Long Journey Through Lysosomal Storage Diseases. Because the title indicates a very long journey. In reality, the journey has been about 50 years long. But I have 30 minutes for 50 years, so I will use some written notes, as you see, to remain on time, I hope. What I will talk about is limited to my personal experience in this field, in which I would like to underline the importance of basic research, translational research, clinical research, which in the 50 years that I have witnessed generated important scientific results thanks to the activity of researchers and scientists and clinicians from all over the world. Unlike what is usually done, also for reasons of time, I prefer to immediately give you the message to take home, just a single message. It's this one. Scientific advances in the last decades allows great hopes and great expectations for the future of diagnosis and innovative therapeutic strategies in the field of lysosomal storage diseases. I would say LSDs because it's much shorter, both for patients and their families. And I want to share with you Albert Einstein's view: learn from yesterday, live for today, hope for tomorrow. The important thing is not to stop questioning. And this is actually the essence of the scientific research. I hope there will be time at the end to make some predictions about the future of medicine, and also in our field, in the era of the digital revolution and artificial intelligence. Lysosomal storage disease include more than 70 diseases caused by the deficiency of various functions on the lysosomes, and have an incidence of approximately 1 in 5,000 live births. The improved understanding about the pathophysiology of these diseases has led to the development of innovative therapeutic approaches over the last 35 years. This is a partial list of LSDs classified mainly on the basis of the accumulated material and other materials, besides mucopolysaccharides, sphingolipids, oligosaccharides, and some common clinical features. Thanks to the great research progress, we have identified enzyme and protein defects and mechanism, and finally, the mutated genes that cause each disease. The sequence of phases in the diagnostic definition and therapeutic management of patients with metabolic diseases, including LSDs, traditionally started from the description of the clinical picture, or the clinical presentation, was followed by the search of the metabolic defect and the dosage of the enzymatic activity presumably deficient, and more recently ended with the identification of the gene mutation and the prescription of a therapy. It's interesting to note that subsequent phases of the traditional clinical approach are more or less the same as those that progressively led to scientific discoveries to the current state of our knowledge on the pathophysiology of LSDs and to innovative therapies. in traveling with you on this journey through the various stages achieved thanks to research, I will use what has been published by Giancarlo Parenti and Andrea Ballabio in this article. Giancarlo Parenti and Andrea Ballabio are the two young people portrayed in these old photos who became then professor of pediatrics and genetics at my university, Federico II University of Naples, after having been in my group as students in the medical school and then at the of the residency in pediatrics. The figure from this paper, from this article, illustrates the evolution of knowledge in LSDs, which is what I have also seen on my journey as a witness. In the 19th century, the first phenotypes of what were later classified as LSDs were Tay-Sachs, Gaucher, Fabry, Niemann-Pick disease, type C, although at that time everything was not so well known. But Hunter syndrome was described in 1917 and Hurler syndrome in 1919. And it seems strange that in this photo, two brothers with Hunter syndrome are described in the only chapter dedicated to Hurler syndrome in a book of the late 50s by the most famous clinical geneticist at that time, Victor McKusick. But I will talk about Victor McKusick later. So the journey begins with the identification of the clinical pictures without yet knowing what this disease were due to. Only in 1955-56, lysosomes were identified by this scientist, Christian de Duve, who won the Nobel Prize in Physiology and Medicine in 1974. And I had the chance to attend a lecture of his soul only once in my life. In the years 63-70, the enzymatic defects causing LSDs began to be identified. In this period, my first contact with this disease started. In 1969-71, as a biochemical research fellow, not as a pediatrician at that time, I was at the National Institutes of Health in Bethesda in the laboratory of Chris Sanvinsen, who won the Nobel Prize for Chemistry in 1972. And in a series of scientists of various backgrounds worked in his lab in decades, in 10 years from 1965-75, and I just underlined in red those who were coming from my university in Naples. Of this, I want to remember Paolo Di Natale, later professor of biochemistry, with whom I had a long collaboration in the field of LSDs in Naples, since she had also a further internship at the NIH in the laboratory of Elisabeth Neufeld. During my stay in Bethesda, I had the opportunity to listen to seminars by Elisabeth Neufeld and Roscoe Brady, in which they illustrated the results of their discovery. Brady had begun to identify the enzymatic defects of some diseases, namely Gaucher, Niemann-Pick A and B, Fabry, and Tay-Sachs. And Elisabeth Neufeld identified those MPS 1 and 2. And the key experiment of Elisabeth Neufeld, she was talking to the audience, started as an accident in the mixing of cells of different genotypes that resulted in correction of their biochemical defect. This serendipitous experiment led to the identification of the enzyme deficiency in both Hurler and Hunter syndromes, to an understanding of the biochemistry of lysosomal enzymes in general, and to the cell biology receptor-mediated endocytosis and targeting to lysosomes. And this paved the way for the development of enzyme replacement therapy with recombinant enzymes, as we will discuss later. My travels abroad resumed in 1977-78 with an internship in clinical genetics and research at St. Louis Children's Hospital of Washington University in the genetic division directed by William Sly. Sly had described the new mucopolysaccharidosis, which later took his name, MPS 7, or also called Sly syndrome, due to the beta-glucuronidase deficiency. This was the paper, and this is just a slide to remember my look at that time. And at the same time when I was in St. Louis, in the same lab where I was working, there was Marvin Natowitz, just the bench close to me, next to me. And Marvin then discovered MPS 9 due to hyaluronidase deficiency in 1996. So at that time, Sly, former discoverer of MPS 7, and Marvin, future discoverer of MPS 9, were working in the same lab. And these are two personal pictures of our families, with Sly family and my family during time. In reality, at that time, Sly's most important scientific contribution was this one, to identify and clarify the mechanism by which the cross-correction of enzymatic defects and metabolites accumulation is possible in the cultivation of different cell lines with lysosomal diseases, according to the suggestion of Neufeld that lysosomal enzymes contain a recognition marker for uptake and transport to lysosomes. Sly laboratories identified the mannose 6-phosphate as this recognition marker attached to the protein molecules of lysosomal enzymes that binds a mannose 6-phosphate receptor on cell membrane and on that of lysosomes. So mannose 6-phosphate is a kind of identity label present on the mature enzyme, which recognized by the specific receptor allows entry into cells or lysosomes through their respective membranes. Okay. In the 80s and 90s, the molecular basis of many lysosomal diseases were identified, and a large portion of the genes coding for these enzymes was cloned. However, many things remain to be clarified, despite the progress of knowledge on the clinical, enzymatic, and finally molecular characteristics. For example, the enzymatic defect was seen to cause different clinical presentation, what is called the pleiotropism by the geneticists, as in the case of MPS 1 and MPS 2, severe and mild attenuated form of MPS 1. And also for other lysosomal diseases, there is the same situation, Niemann-Pick, AMB, and Gaucher type 1, 2, 3. Also, on the other hand, different mutated genes could cause similar pictures, and this is true for MPS 3 or Sanfilippo disease with 4 defects, and with Morchio disease, where 2 defects have been described. And even the same molecular defect could cause very different clinical presentation, as in the case of these siblings that we described with Paolo Di Natale, affected by mucopolysaccharidosis 3B, who are different about phases, but also about biochemical abnormalities like urinary mucopolysaccharides and so on. So, clinical pictures of genetic syndrome of the type of LS, these were also known. Excuse me, this is the right slide. And there were some LSDs with multiple defects of lysosomal enzymes. This was not compatible with what we know, that lysosomal diseases are monogenic diseases, due to mutation of a single gene. An example is multiple sulfatase deficiency, which on a clinical level recapitulates the phenotypes of various MPS, each one primarily due to mutations in genes encoding specific sulfatases. The genetic basis of the multiple sulfatase deficiency was clarified in 2003 by 2 groups, a German one led by Kurt von Figure and the Italian one led by Andrea Ballabio, who was and is still director of the Teleton Institute of Genetics and Medicine in Naples, and at that time was returning to Naples from the United States. And 2 articles by the 2 groups were printed in succession in Cell, prestigious journal, scientific journal, at the conclusion of very epic competition between the 2 groups. Instead, I would like to tell you as briefly as possible about the other example of multiple defects, galactosialidosis. And here we come back to Victor McCusick, was, I said, the most famous clinical geneticist in the last century. In 1988, Giovanni Romeo, an Italian geneticist, organized a medical genetics course in Sestri Levante, and I was in the faculty for this with, of course, Victor McCusick, that was the lead of the group, and also the young Andrea Ballabbe was present at that time. And when I made my presentation in the presence of Victor McCusick at this meeting, I was photographed with the microphone in one hand and the other hand shaking for the emotion. In 1969, Victor McCusick had written this paper, this article, to clarify how the classification of genetic diseases sometimes require a long process made up of merging and separating disease categories that he called and are called lumping and splitting. And he published this cartoon in this article with 2 characters from American history. You can recognize Lincoln, but I took the liberty of changing the faces with, of the lumper and the splitter with these 2 more recent US presidents. So, what is the history of galactosialidosis? It starts from this Italian child, Raffaele, who had all the characteristics of lysosomal storage disease, but with normal intellectual development. At the age of 18 months, we described him as an atypical case of beta-galactosidase deficiency. At that time, this deficiency was known as the primary cause of 2 diseases, GM1 gangliosidosis and disease type B. But actually, the clinical presentation of Raphael was not at all those of these 2 known LSDs. And we asked the laboratory in Rotterdam, led by Hans Galliard. They verified that the fibroblasts of our child and other atypical cases of beta-gal deficiency complemented, were cross-corrected with those of GM1 gangliosidosis. So this ruled out that this beta-gal was not the primary defect in this child. Then, during my stage in St. Louis, in the genetic unit directed by Baslai, I took care or looked after this girl, Natalie, with characteristics similar to those of Raffaele and an atypical beta-gal deficiency again, but with also normal intelligence. Meanwhile, a new lysosomal storage disease was discovered caused by the deficiency of neuraminidase and sialidase, new LSD, that was called sialidosis. Neuraminidase activity was assayed in our 2 patients, and also deficiency of this enzyme was found. So the diagnosis seemed to be sialidosis. However, complementation studies in the same laboratory in Rotterdam ruled out this possibility because there was a complementation of Raffaele's fibroblasts with those of primary defects of neuraminidase. Finally, to make a long story short, still in Hans Galliard laboratory, thanks to the Italian Alessandra Dazzo, a scientist, the primary defect of this disease with these 2 enzymatic deficiencies was identified, and the primary defect was the deficient production of a so-called protective factor, which protects the molecules of the two enzymes, beta-galactosidase and neuraminidase, from degradation that leads to their secondary deficiency. And the conclusion was that the primary defect is deficiency of another protein, that is this protector function with also an enzymatic activity called cathepsin A. What was my contribution on this story was just to help in classifying three types of these patients that have been described in the world, and to coin this name, galactosialidosis. This name was then accepted by the McCusick catalog of Mendelian disease, and also in this classic textbook of metabolic and the molecular basis of genetic diseases, that is now the Scriber's book known, and there is a chapter with Sandra Datz in the first edition also with Hans Galliard describing this new LSD. And these are some pictures in 1990 in Rotterdam with also Galliard and his wife and Giancarlo Parenti and Sandra Dazzo here, and this at lunch with Hans Galliard and our wives. So the history of therapeutic approaches to LSD begins, and this was due to introduction of the enzyme replacement therapy, but also other approaches were found. In this cartoon, we see that the gene mutation can lead to the synthesis of mutant enzymatic proteins, which may undergo rapid degradation, therefore lead to a deficit of these enzymes, and this, as a consequence, cause the accumulation of non-degraded material in the lysosomes, but also to progressively secondary abnormalities in the cell. So the enzyme replacement therapy was the first approach, and I don't want to talk to this because it's very well known, but there are other approaches like substrate reduction, pharmacological chaperones, and gene therapies that progressively were found. The inhibition of substrate synthesis is a strategy aimed to block the production of the substrate and that accumulates and therefore compensates for the lack of degradation due to the enzymatic genetic defect. In other words, if the degradation of the substrate that comes from the outside or that is synthesized in the cells is insufficient due to the enzymatic defect, we try to inhibit the synthesis of the substrate and balance the degradation defect, thus reducing the final accumulation of the lysosomes. Let's go back to... this slide shows a recurring name in this approach with substrate reduction therapy, to reduce substrate synthesis, sorry. This name is miglustat, a drug that was initially used for Gaucher disease as an approach to inhibit the synthesis of glucosylceramide, that is the substrate that accumulates in this disease. This step, a crucial step that is blocked by miglustat, is also precursor of many other steps that are involved in a series of lysosomal storage diseases that are indicated in the slide in green. Okay. One of these diseases, the Niemann-Pick disease type C, and there were trials using as an inhibitor of substrate miglustat for strategic treatment of these diseases. at the beginning, Dr. Pinedo was involved in this, one of these trials sponsored by the industry. We in Italy had the trial supported by the Agency of Drugs. The Italian agency was independent, and the results were actually encouraging. And still, microstat is the only drug used, approved at the moment, but many research are going on for other innovative drugs. The pharmacological chaperones is a small, another possibility was to use the pharmacological chaperones to increase the amount of enzyme available for substrate degradation. The goal is to improve the so-called tertiary structure and stability of the mutant enzymatic protein. When protein is synthesized, there is a quality control of this protein that is possibly degraded if it does not work well. And here, this is the aim to use pharmacological chaperones, small synthetic molecule that helps stabilize this tertiary structure unstable by binding to the mutated protein, correcting and stabilizing its structure, and accompanying it until intended lysosomes. Many of these studies on pharmacological chaperones were conducted in our group by Giancarlo Parenti, who identified the particular property of one of these molecules, and again, miglustat, which in fact we have already encountered for the substrate inhibition approach. Parenti first documented that miglustat is also a chaperone of the mutated enzyme alpha-glucosidase in Pompe disease and increases stability and activity. But he also later discovered that the same chaperone is also able to improve and increase the activity of the recombinant enzyme that we thought was just exactly the same as the native normal enzyme. The recombinant enzyme that we used for the enzyme replacement therapy. We carried out a first clinical trial on the combination of ERT, of enzyme replacement therapy, and chaperone in Pompe disease, and showed an enhancement of blood alpha-glucosidase activity in patients treated with enzyme replacement therapy plus chaperone compared to enzyme replacement therapy alone. And these pharmacological chaperones are becoming widespread now, strategies in LSTs, and also the combination of chaperone with recombinant enzyme has been developed, recently approved by the pharmaceutical industry in Pompe disease. But also in Fabry disease with migalastat. I have left for last what would logically be the first therapeutic approach for lysosomal disease, namely the correction of the genetic information that is now possible with so-called gene therapy, or genome editing, antisense oligonucleotide, and so on. The two strategies for gene therapy are the replacement of the defective gene with the normal gene, and more recently, the genome editing, a tailoring repair of the gene defect. In 2020, these two bright scientists were awarded by a Nobel Prize in Chemistry for the development of the CRISPR technology that is used for the genome editing and that is in progressive development for clinical application. For gene therapy, classical one, first approach is the in vivo one, with the administration of a correct gene, usually through a viral vector, which colonizes, for example, liver cells, thus transforming these cells into a sort of factory of the normal enzyme, which is then spread through the bloodstream to all organs and corrects lysosomal storage. There is also the ex vivo technique, with which hematopoietic stem cells are taken from the bone marrow of the patients, cultivated in vitro, and transduced, corrected with the vector containing the correct copy of the gene, to then be auto-transplanted to the patients without risk of rejection. With this strategy, the marrow is transformed in the factory, not only of the enzyme produced by the transplanted cells, but also in the factory of the same cells that can migrate from the marrow through the circulation and can pass the blood-brain barrier and enter into the brain. It is important to remember now how useful international collaboration in research projects has been for the advancement in knowledge. I would like to remember this grant financed by the European Union, where my contribution was first to invent the acronym Euclid. In this grant, the institutions involved were two from Italy, Naples, the Netherlands, UK, Lund University in Sweden, Germany, Gutenberg University. The aim of this project was to study lysosomal diseases without neurological involvement, going from the laboratory to the bedside. And the aim was to study pathophysiology and natural history, investigate various approaches for the treatment of these diseases, from enzyme replacement therapy to substrate inhibition, chaperone, and gene therapy. The studies diseases without neurological involvement were primary neurological involvement, were Gaucher disease type 1, mild MPS 2, MPS 6, Pompe disease, and multiple sulfatase deficiency. This slide shows a recurring name among drug, but the project that most involved us in Naples was that of the clinical trial for gene therapy MPS 6, which finally moved from the preclinical level to the clinical level. And the trial was again called MUSIX on the basis of my proposal. The clinical centers involved were Federico II University in Naples, Ankara University in Turkey, and Erasmus University in the Netherlands. Alberto Riccio was the coordinator. There were various collaborators. I also collaborated before my retirement, and now Nicola Brunetti Pesi became the coordinator for the clinical part until the conclusion of the trial. And now I am pleased to record that there was an ethical advisory group that had been established, chaired by Professor back in Oslo, and including heads of patient associations from Turkey, Italy, the United States, Spain, France, the Netherlands, the United States. And we see them in a meeting that took place in Rome in 2014. And of course, you can see very well Jordi Cruz as a representative of Spain. But I would like to address a thought to Christine Lavery of the United Kingdom, who unfortunately passed away in 2017. And I remember well the last occasion when I met her in Madrid during a previous conference of the MPS Association, to which I had been invited. The results of this trial, the MPS 6 gene therapy, has been published in this journal, New England Journal of Medicine Evidence in 2022, and this means that from the granting of the financial support from the European Union to the publication of this final report, practically 10 years have passed. So in this trial, 9 patients were recruited and treated in cohorts with 3 different doses of the drug, and the fundamental results, very briefly I will tell this, are summarized in the summary, in this abstract. So the gene therapy direct at the liver, so the in vivo approach, demonstrated, especially the highest dose, the dose with the highest concentration of drug, a stable enzyme activity between 30 and 100% of normal average activity. And also was seen only a modest increase in urinary mucopolysaccharides, which did not reach the concentration for which a reintroduction of enzyme replacement therapy was necessary. Patients who received the highest dose did not show clinical deterioration during observation for up to 2 years following the implementation of gene therapy. The last important aspect of this discussion of the knowledge of lysosomal diseases is what happened from 2008 and 2009 onwards on the identification of autophagy defect, but above all on the identification on the transcriptional control of the biogenesis of lysosomes and an actually revolutionary characterization and understanding of lysosomal biology. Andrea Ballabio played an important role in this, and it is no coincidence that this masterful reading in 2016 at the Dewey Institute in Brussels was entitled "The Lysosome from a Waste Bin to a Control Center for Cellular Metabolism." I don't want to summarize the results of the research made by Ballabio's group, but in 2009, Ballabio's group discovered this master gene called TFAB, which controls the activity of lysosomes. And the gene acts as an orchestra conductor, regulating the expression of many other genes involved in the clearance of materials derived from cell metabolism and in energy metabolism. TFAB mediates the cleansing of accumulated materials from cells, not only with lysosomal storage disorders, but also probably with neurodegenerative diseases like Huntington's disease, Alzheimer's, and so on. And this is a cartoon a little bit old, 2016, I think, where there is a summary of what kind of other research in other fields this discovery of TFAB causes. And I want to emphasize what I said, that some of these results have been applied, at least in the lab now, for these neurodegenerative and common diseases, actually. Also, the secondary cellular anomalies can be influenced by this kind of discovery, and for example, on exocytosis, the expulsion of material from the cells, and secondary correction of abnormalities in the cells, like those of autophagy. The last 3-4 slides, and I am at the end of my long presentation, are about the future that I will not see, of course, but many of you will certainly see. I have realized how important was the statement made in 1997 by Francis Collins, who was the coordinator of the so-called Human Genome Project that sequenced the entire human genome, and was then director of the NIH from 2009 to 2021. Collins stated virtually all the pathological conditions that medicine deals with have a genetic basis, or at least a genetic component, perhaps with the sole exception of traumatic pathology. From this statement, the foundations are derived for what the new medical scenarios can be, considering what scientific progress has already achieved. First, in these new scenarios, we will see, so these are prospective prevision, all neonatal or even prenatal screening for genetic and metabolic diseases will be increasingly based, more than at present time, on molecular analysis, not only limited to the exome, that is the small coding portion of DNA, but will be extended to the entire genome, including parts of the genome whose function we do not know yet. Neonatal screening will allows us not only to identify the pathological variants of rare monogenic diseases, but also genetic variants that may play a role by interacting with each other and with non-genetic and environmental factors, and determine the clinical pictures of common pathology. I mean, the most frequent causes of mortality and morbidity, heart disease, epilepsy, metabolic diseases of the adult. So these polygenic risk scores are being studied and calculated with the help of the artificial intelligence and machine learning. Therefore, a genetic diagnosis of monogenic diseases and also these risks for common diseases eventually presenting at later ages will allow interventions with very early innovative therapies like gene therapy, but also follow-up to control lifestyle and prevention based on the risk factors identified. As a matter of fact, the future of medicine and of society in general has already begun, and is based on artificial intelligence and machine learning to achieve what is the current goal of the so-called precision medicine or personalized medicine. Artificial intelligence helps to analyze an enormous amount of data deriving from the most varied documents of our daily life. Therefore, the organization of healthcare will be revolutionary on the basis of this new technology for diagnosis, for personalized therapeutic programs, and for therapeutic technologies protocols. And these products will be increasingly tailored to the individual patients and managed by algorithms. So this is a summary of what we can expect. These new laboratory technologies, genomic sequencing on newborns, personalized therapeutic programs, gene therapy with one-shot therapy and hopefully lasting effectiveness, and then artificial intelligence and machine learning to manage these personalized protocols and produce algorithms. And so a new organization of healthcare. Finally, this historian from Israel, Yuval Noah Harari, is the author of various bestsellers, I should say. The last one is the 21st lesson for the 21st century, in which he makes a provocation stating, if someone describes a mid-21st century world to me and it seems like science fiction to you, it's probably false. And then he added, but if someone described the world to you, the world in the middle of the century, and it doesn't seem like science fiction to you, it's also certainly false. In this world increasingly dominated by artificial intelligence, even in medicine, we will have probably a robot that sets us for a checkup appointment in the clinic, and perhaps it will be our interlocutor during the visit. So a pessimistic conclusion might be, while we easily talk about personalized medicine, are we moving toward the goal of depersonalized medicine? We started on this journey from the title of film about the way we were, and we arrived at a conclusion, which is the theme of the song from Casablanca, Time Passes. Well, it was also possible to hear just the beginning, but I don't know if we are technical in this possibility, it doesn't matter. But you probably know this movie, this song. And to conclude, I want to give my sincere thanks at the end to all personalities I have been lucky enough to meet and whom I mentioned during my presentation. However, I particularly thank Giancarlo Parenti, also the author of many of the artistic slides I showed, and Andrea Ballabio. From both of them I have learned much more over the course of several decades than I have perhaps managed to convey to them. With this optimistic image of a rainbow framing Vesuvius in the Bay of Naples, despite the defeat of the Napoli team in the match against Real Madrid, I thank you very much for your attention. Thank you.